You can tell it kind of bothers me. Yeah. Chapter 14, the signal transduction pathways. Woo! Making moves. Such a great. This is straight up from your book, and it is so, what a wonderful use of art and science, right? Everybody see the signal transduction causing somebody to scream and be overwhelmed. Painting being, if I remember correctly, titled Scream. Oh, man. Sometimes it's those little attentions to detail that make it really worth actually looking at your book. Okay. So the many signal transduction pathways share common themes. The signal transduction pathways, the chain of events that convert molecular messages into the range of different physiological responses. Your transduction is the conversion of the information of the process or the concentration of a signal molecule into other forms. Notice again, signal transduction pathway, the chain of events, right? Then we have the individual transduction, okay? Which is the event, okay? Singular event. The signal transduction pathways are going to contain certain key steps. The release of your primary messenger. Another way of saying that is the signal, right? This is the signal that is going to be transduced through the pathway, okay? The reception of your primary message by a receptor. This is going to be a membrane protein with intracellular and extracellular components because it has to transduce that message through. The delivery of the message inside the cell by the intracellular second messengers. So you have a primary messenger binds to a protein. The protein is going to cause some sort of secondary messenger within the cell. The activation of other molecules that directly alter the physiological response. The termination of your signal. Okay, so I know that we're doing this in a very broad, which I normally don't do such a broad format for these introductions, right? Usually we get into a more specific right away. The reason we need to do a very broad format is there's actually multiple of these types, okay, that have some pretty significant divergences in their specifics, but they always fulfill these broad steps, okay? So it really will help you remembering what you should have done with these other types, okay? And what do they mean? Okay, one of the important things about what I just mentioned is every step of these, when we have enzymes or proteins involved in these steps, what can happen to those steps? What happened to our cascades every step? Can an enzyme just do it once? No, an enzyme does it how many times? Can do it quite a few times every step, right? And so when we get something where we have an initial signal, it then has a response. That response can be much bigger than the initial signal, right? And then you have something that responds to that and gives a further signaling. That means we have a further amplification, right? And so we again have a rapid cascade here, okay? So we can have signals such as a change in hormone levels. This can lead to responses through a signal transduction pathway, which we're seeing right here. And so we have our initial signal hits our receptor. So our reception here. We then are going to have amplification. Now these amplifications can happen multiple times or they can just happen once. Depends on how drastic you want your response to be, right? If you have a single amplification, it's going to be a outsized response for the signal, but not ridiculous, right? But if you have multiple amplification pathways, it can be very large responses for very, very tiny amounts in your blood, okay? Which is a lot of steroids, right? The amount of like caffeine you have in your blood, the amount of like cocaine in your blood that takes to activate something, the amount of testosterone or estrogen, not a lot, because there's multiple amplifications. So transduction is going to be the next step after your amplification, and then you'll have whatever your responses are. Now, these responses can actually branch within a single cell, but they could also be different and have multiple branches for different types of cells. Okay? You may have noticed this for like things like insulin and stuff. It has different things that it does in different types of cells, right? Have you guys gotten into any of that? Insulin, what does? Okay, okay. So we're starting to hit things that you haven't talked about. Cool. Fun. So these are your common secondary messengers. In this list right here, we probably have something like 80% of secondary messengers. Okay, maybe more. I never have done the math, but it's the vast majority. Right here we have, say, CAMP or CGMP. Okay, this is either an A or G here, and this is the AMP version or the GMP version. Notice the C here. What does the C tell you? Cyclic. Yeah, cyclic. Okay? Single phosphate cyclic. This is the calcium ion right here. Notice this is the hydrated version because in the cell it's going to be the hydrated version, right? Okay, this is the IP3. Have you guys ever heard of IP3? No? Ooh. Okay, I'm getting to break lots of stuff out to you. How about DAG? No? Okay. Okay, so this is DAG right here. This is diacylglycerol. Notice the glycerol right here, 2 acyl groups. Okay, and this right here, IP3, what do you notice about this? It has 1, 2, 3 phosphates, and it's inositol. Notice that's a like sugar derivative, right? Okay. So what are some of the consequences of secondary messengers? They may amplify the signal significantly. The messengers are often free to diffuse and influence the processes throughout the cell. What do you notice about most of these? That's pretty polar, right? That's pretty polar. That's pretty polar. This is the least polar one, isn't it? What do you think this one commonly ends up being located? What does that look like? Does it look like anything? Like a phospholipid, maybe? Yeah. Yeah, so you end up seeing this a lot in like the cell membrane. Okay? Typically, this actually gets a part cleaved off, usually one of these. And it floats around the cell membrane, the other one floats into the cell and works on the insides of the cell. Anyways, it's cool, right? The function is dictated by what? The structure, right? Yeah. Okay, so again, a lot of these messengers are going to be free to diffuse and influence processes throughout the cell. Again, how they diffuse dictates where they actually will activate things, where they'll work. Use of common second messengers in multiple signal pathways does create crosstalk. Yes, that is a problem. That is an issue. That is a, what's a weird thing that happens but it's not an issue? It's not a problem. Consequence? Does that make sense? Okay. It's not always a bad thing. Okay? Sometimes the crosstalk actually creates things that are necessary. So these are 3 different pathways that are going to illustrate many of the principles of your signal transduction. Keep in mind though, this is your overall signaling dynamic for basically every hormone that you can think of. Okay? Like we just stepped through a big door into a huge warehouse of stuff. All right? This was one of those moments where you're going to go back and go, oh my God, Dr. Clausen, you just made me just open this door to all these items I have to deal with. Okay? Okay. By the way, any medical person, this is the stuff that really is where you cause a lot of problems, okay? That you're going to really have fuck-ups from people and you really should be aware of. So when it happens, you can go, oh, let's try this. Like 30% of patients are going to experience some sort of problem with their medicines, will have been given the wrong medicine or the wrong amounts. And of those patients, many of those die. Okay? It's called the drug spiral or the death spiral. And it's doctors and PTs and nurses and PAs and everybody that understands these and goes, hey, maybe we should shift that off. Don't keep assigning more and more drugs to them. That'll just kill them. Okay? Okay, and this is coming from a chemist. I like my drugs. So this right here is the epinephrine. This is the beta androgenic receptor here that the epinephrine binds to. And so we have some pathways, amplifications. These are the secondary messengers we were talking about, and that signal transduction we were just talking about. And this is going to cause overall the muscle contraction or relaxation. Okay? This is the insulin plus the insulin receptor. And again, we have some processes here. These are some pathways we'll talk about more in detail later. And this is going to cause, in this case, increased glucose uptake. This right here is the EGF, the epidermal growth factor, the EGF receptor, and then we get expression of our growth promoting genes. Okay. And notice that we all have a very similar pathway, right? We're going to go through some sort of secondary messengers and cause some activity in that cell. Okay, so here's the epinephrine signaling. This is going to be a heterotrimeric. G protein transmits signals and resets themselves. So this will be the androgenic receptors that are going to accept this signal. This is the cell surface proteins of two classes. We have the alpha androgenic receptors and the beta androgenic receptors. The epinephrine, another name for the epinephrine being adrenaline, maybe the one you're more familiar with. Okay. Though I guess epinephrine pens, you're probably familiar with that, right? Yeah. So same thing. Though that's usually pre-epinephrine in the epinephrine pens, but anyways, not really important. The hormones secreted by mammalian adrenal glands in response to the internal and external stressors. It's going to exert the fight or flight response typically. That's what most people associate that with, the adrenaline part at least. And it binds to the beta androgenic receptors. So there's 7 transmembrane helix or 7TM receptor. The 7 transmembrane helix, I'm going to say 7TM receptors from now on. Is everybody going to be okay? How many of them that go through the membrane, by the way? 7. Nice name, right? Yeah. This is going to be a large class of cell surface receptors, okay? We're going to see many of those. The transmit diverse information initiated by hormones, neurotransmitter, odorants, and light. There are going to be nearly 1,000 that are coded in the human genome alone. Okay? Don't worry. I know you're having a heart attack right now because of your fight or flight response, but I don't make you memorize all 1,000. Okay? Just 999. No, I'm not going to make you memorize a lot of these. But basically, if you know the pattern for 1,7TM, you basically know the pattern for them all. You okay? That flight and flight response, the adrenaline going through you causes shakes, right? Muscle contraction issues. Yeah. Okay, so frequently therapeutic drug target. Basically, if you have any drug where you can take like a less than 1 milligram doses into the microgram doses, then it's probably a 7TM target. Okay? This contains 7 helices that span the membrane layer, obviously 7TM. This is actually what this looks like, again, in abstractia. Okay? So this right here is the N, that's the amine side, right? And then we loop down 1, 2, 3, 4, 5, 6, 7, right? And then we get to the C side. Okay? This is the inside of the cell. Outside of the cell has the amine. Inside of the cell has the carboxylate. Okay? Notice it just straight up anti-parallel every time. Neat, huh? Okay. Though do note that these are not beta. I said anti-parallel because they're opposite sides, right? Yeah. They're alpha helices. That's why they look like rods. So the three-dimensional structure for your beta androgenic receptor looks like this in abstraction. The ligand binding sites for your ligands is going to be toward the center of the bundle of helices, about 20% of the way into the receptor from the extracellular site. So this is, again, the extracellular side here, intracellular side here. And again, that's the beta-2 androgenic receptor. And what you can see is that, yes, it is not actually flat in the membrane. Okay? It does make a barrel shape. It's actually wrapped in on itself. But you couldn't have seen the nice wrapping like this, right? If I'd had it like this first. You can kind of see it, but it's not obvious. See that? You can see some of them. Anyways, this right here is what this looks like if you look top down here. And again, you can see the directionality of this. 1, 2, 3, 4, 5, 6, 7, like so. Okay? It's literally wrapped around in a little spiral. Okay, so this is the binding right here. This is binding of an agonist to the beta androgenic receptor. It's going to result to your stabilization of the activated form conformation of the receptor. So agonists are going to be ligands that activate receptors. It can be a variety of things. It just means agonist like a protagonist, right? Okay, activates it. Okay. So again, this is going to activate this receptor. Notice this right here. Your helices have actually moved. Can everybody see that okay? Okay. So specifically your helix 6 and helix 5. 5 and 6. Everybody see those okay? Those moved. Okay. Those are the major ones that move the most. They do the most binding. Upon binding. They move the most upon binding. Okay. So your ligand is going to bind to your 7TM receptors. It's going to lead to the activation of your heterotrymic G proteins. So your conformation change on the cytoplasmic side of your receptor is going to activate your G protein. Which side was this? Yeah, cytoplasmic. Yeah. See, okay? Those are back there. Okay. All right, so again, your conformational change on the cytoplasmic side of your receptor activates your G protein. This is going to be the next protein we're going to be starting to talk about. That's that heterotrymic, G protein. When activated, your G protein is going to stimulate the activity of the adenylate cyclase. What do you think the adenylate cyclase acts on? Adenine, A, cyclic, CAMP, right? Yeah. So this is going to be the enzyme that's going to catalyze the conversion of ATP into CAMP. Now, CAMP was one of the common secondary messengers, right? Yeah. And when you see CAMP or CGMP, you really should start going, ooh, we may be talking about a secondary messenger. We're probably going to see some amplification. It's going to activate something, okay? So your secondary messenger, CAMP, this is going to carry your signal throughout your cell. It's highly water soluble and it can move fairly easily throughout the cell, okay? Notice that, is this going to just do once? Are we going to have an enzyme that just goes, oh, 1 ATP to CAMP, I'm done? No, so we're going to have one molecule, the agonist, that binds. This is going to activate an enzyme that's going to make many CAMPs fairly rapidly, right? Okay. And so one molecule binding had an amplification effect. Mm-hmm. The agonist is going to be the one that will activate your receptor. So yes, it can be the primary messenger in this case. Okay, so here we have the G protein, by the way, down here. This is why it's referred to as the G protein. It's GTP, and then it converts Okay, so right here again, you have your epinephrine. This is going to be binding to the beta androgenic receptor. Notice, by the way, the coloring matches here. So if you see here, the binding of the agonist turned it green, i.e. on, go. In this case, our agonist is epinephrine, because we're talking about specifically the beta androgenic receptor. Okay, you can also see the sites here if you match them up to the previous figure, but because we went to green in this figure, it doesn't have the nice color coding that the previous figure before that other one did. Okay. Anyways, this is the G protein right here. And so what you're going to see here is you're going to have this activate. It's then going to work on the G protein. This G protein then is going to send off this portion right here with the GTP bound to it, to the adenylate cyclase. The adenylate cyclase is the one that's going to start activating the ATPs and making CAMPs. Okay? And it's going to keep doing that until it stops. Okay? And that means that one molecule is going to have many, many of the CAMPs activating the next part. Okay. Now, these actually remain attached right here, this G protein and adenylate cyclase. These remain attached to the membrane here, okay? So those actually don't end up detached from the membrane, okay? Now, the CAMP is the one that's going to drift off into your cell. In this specific case, by the way, it's going to be part of the process of activating protein kinase A. So we'll activate protein kinase A from its inactive form, okay? So in its inactive form, the G protein is going to consist of a heterotrimer. And so right here, we can actually see that heterotrimer. Hetero meaning different parts, right? Trimer, 3. Okay. And so we're going to have alpha, beta, and gamma subunits. The alpha subunit, which is G alpha, this is going to be bound to the GDP. And this is this one right here. Everybody say okay? The alpha subunit. Very good. The alpha and gamma subunits are going to be anchored to the membrane. This can be done by covalently attached fatty acids. So these are the parts that are anchored down. You can actually see the covalently attached fatty acids here, by the way. Can everybody see those little black things that are mixed in? Yes. Yeah. Okay. And those are the covalently attached fatty acids. That'll keep those anchored down. This is actually what this looks like in a less abstracted form. And so right here is our alpha here. Remember that, see, you can actually see the slight purple here if you pay attention really, really close. It can be a little confusing before you notice that. But this right here again is that GDP binding alpha unit right there. Okay. In whole. Then this is a different subunit, then this is the other subunit. Okay? Okay. And that's there right there, without all the other stuff. Okay. Now, this is the gamma subunit again. Notice again, it's going to be anchored onto the cell membrane, right? Alpha subunit, that's anchored onto the cell membrane. Okay? This is the one with the GDP. See, it's closed because it has the GDP. This is then the beta subunit right here. And so here we have the gamma, beta, and this right here being the alpha subunit. This is actually the GDP right there. Okay. Right there. All right. So the G alpha is going to interact with the surface of your receptor. This is primarily the helices 5 and 6. Which ones were the ones that changed the most? 5 and 6, yeah. What were those literally part of? Those were literally changing how this subunit here binds to GDP and GTP, right? Like they're little pistons. They're opening and closing doors, right? Yeah. Okay. And so when it's bound to the receptor, the G alpha is going to open substantially. This is going to enable your replacement of your GDP with GTP. Okay? And so when it's open, GDP can drift off and GTP can take its place. When it's closed, the GDP will be trapped. So this is the whole setup right here in a little bit more detail. And so here again, we can see the extracellular side of the 7TM, cytoplasmic side, and here it's connected to the G alpha here. Here we have the beta and gamma. Right here, we actually have these labeled as G, beta, and gamma together. Okay, we're not separating them right now. We're not talking about them as separate subunits. Okay, so right here, this is the G alpha S, singular. This is going to have your GDP form in it, and here's your bound nucleotide actually right here. This is the G alpha S with the receptor bound right here. And you can see that it's cracked open. Everybody see the little lid here? Fantastic. Okay, that was that gray part that cracks open and allows you to remove the GDP and replace it with GTP. Okay, so on GTP binding, the G alpha is going to dissociate from your beta gamma dimer. That's why those are referred to as the beta gamma, the G beta gamma subunits right here, whereas this is referred to as a singular subunit by itself. Okay? So those are going to separate upon binding to GTP and from the receptor. It will dissociate from the receptor as well. So this is binding GTP, disassociates here, disassociates here. The receptor is then going to be available to bind other G protein heterotrimers and catalyze the exchange of GTP for GDP. This is going to again be another level of amplification. So once this is unbound, this is drifting off and going to activate adenylate cyclase, right? But this is going to leave the 7TM still there, right? So what can then happen is another one of these setups can assemble on here. It will then allow exchange of the GDP to GTP. Once exchanged to GTP, it can go off and bind another adenylate cyclase, right? And so we can do that over and over. Notice that arrow, arrow, arrow. We're getting there, aren't we? We got arrow, arrow, okay? Okay. The G protein coupled receptors, abbreviated total GPCRS. I've never seen somebody that's so ridiculous. They would abbreviate that. Usually they would just leave it, the G protein coupled receptors, and not try to abbreviate that further. Because this starts getting confusing when you have that many abbreviations. So this is another name for your 7TM receptors because they signal through G proteins. So you can again call these 7TM receptors a G protein coupled receptors because their signaling molecule is the G protein, right? That's technically the secondary messenger for this one, okay? And then it will do another messenger, which is your secondary messenger, CAMP. Okay, so the adenylate cyclase, this enzyme that converts ATP into CAMP, it's going to be composed of 12 membrane-spanning helices. The catalytic part is going to be made of 2 cytoplasmic domains. In the beta AR pathway, this is going to be activated by G-alpha, or G-alpha S, which has the substrate bound to it. This is going to bind the adenylate cyclase. Binding is going to favor a more catalytically active conformation of the adenylate cyclase. This is going to provide another level of amplification. By the way, notice the more active, catalytically activated form. Okay? Meaning this was partially activated before, right? Many of these processes and enzymes, unless you completely destroy them and scrap them, they'll have some activity. It's actually kind of hard to get rid of all activity of an enzyme, right? as we saw from all the graphs we've done before. Catalytic activity. They never went to zero, did they? Yeah. So this right here is, again, your 7TM right here. I'm sorry, not 7TM. This is your adenylate cyclase right here. That was a Freudian slip again. Notice that, again, you have that very similar sort of anti-parallel alpha helices that are anchoring this along the cell membrane, by the way. In this case, though, it is not 7, is it? Anybody remember how many? 12, yeah. The other thing that separates them, by the way, is the N and C are both inside the membrane. So this right here is, again, the different subunits of this. This here, right here, is actually going to be, that binds over here, is going to be the G alpha. Again, it's just with the substrate, the GTP form of this, okay? And this is what's going to come along. It's going to further activate this. This right here is the actual enzymatic portion, and this up here is the anchor portion. Okay, so let's talk about the next step of this. The cyclic AMP is going to stimulate the phosphorylation of many different target proteins by activating protein kinase A. So the protein kinase A, pKa, have you guys heard of that before? pKa? Okay, so that's the protein that's going to phosphorylate specific. I know you've heard of it here, but I was kind of curious if you heard of it in the other. Okay, cool. So that's the one that phosphorylates the specific SER and 3 residues in target proteins. This can alter that activity. Which way does it alter that activity? Depends on the protein. Okay? It could inactivate or activate. It depends entirely on the protein whether that needs a phosphate there, right? Okay, so that's why it's just alter the activity. This is going to consist of 2 regulatory R and 2 catalytic C chains. And so we have R2C2, again, 2 regulatory, 2 catalytic. Okay. The inactive, in the absence of CAMP, binding of CAMP to the R chains, the regulatory chains again, is going to free your C chains, which are then going to be catalytically activated when freed. And so this is the overall pathway that we just talked about. I'm going to go ahead and define these terms over here, and then we'll talk about the overall pathway. So the troponin complex, the complex found in the cardiac and skeletal muscles that blocks the ability of myosin to bind to actin. This is going to prevent muscle contraction. It'll contain protein troponin 1. In cardiac muscle cells, the pKa is going to phosphorylate troponin 1. The phosphorylation is going to weaken the troponin binding to the actin. And so let's go ahead and see this overall thing here. What was one of the things epinephrine could do? Yeah, it can do fight and flight, but a more specific pathway was the muscle contraction, right? Yeah. And so we're seeing this more specific pathway that I brought up a long time ago. It feels a long time ago, right? We covered some ground. Anyways, this is the epinephrine. Again, we're binding to the receptor, so we have the binding happening here. This is going to give us our activated receptor. If you want to talk this in abstractia, yes, it can be the agonist. Okay. That's just a way of talking about an extraction without having to talk about epinephrine specifically. So agonist and 7TM. This is your activated receptor then. So what's going to happen is we're going to have an amplification step here. That's where you take the GTP that's going to exchange in for the GDP. And this is going to give you your activated G protein. That activated G protein is going to end up forming a protein-protein interaction that's then going to activate adenylate cyclase. I know that biochemistry tends to play a little bit loosey-goosey with steps here. Like, are you guys okay that you understand that we oftentimes skip the protein release parts and that sort of stuff, right? because they're just not very interesting for most things. And they typically happen about speed of diffusion. Okay? Anyways, so this protein interaction, you have the activated adenylate cyclase. This is then going to give you your enzymatic reaction. Again, this is another amplification step, and this will give you increased cAMP within the cell. This increased cAMP is going to activate your protein kinase A. In this case, it's going to be a cardiac muscle protein that's going to be acted upon, that's going to have a phosphorylation, which will then cause contraction. Okay? And so now we have pumped a bunch of epinephrine into somebody's muscle, and they are contracting like crazy now. Okay? Okay. By the way, that's why, have you guys ever seen somebody really go into a fight and flight response that has good muscular definition? No? You guys ever seen like an MMA match? Like a really serious one where they really get at each other? Okay. What happens? Clinch a lot, right? Yeah. That's why. Is the epinephrine is causing more clinching activity. Yeah. That's why they get like super defined, super tight, clinching all over the place. It's not necessarily just them doing it. The G protein is going to spontaneously reset themselves through GTP hydrolysis. By the way, the better fighters will not do that because they'll be more trained not to avoid doing that. You're actually more clumsy when you do that. Yeah. I won't go into why that. So, anyways, did that make any sense? Okay. I don't know if fighting's actually a very good example, but whatever. To me, it is. Which tells you a lot about my life. The G-alpha subunits are going to promote the hydrolysis of bound GTP. This is going to be to the GDP and the free phosphate. This is going to be called intrinsic GTPase activity. The hydrolysis is slow, it's seconds to minutes usually. This is going to allow for that activation of downstream components before it deactivates. So in this case, the cool thing about the G-alpha subunits is they self-deactivate because they actually are causing hydrolysis on their own, right? Okay. This is going to provide an additional opportunity for regulation if the GTPase activating proteins are actually included in the pathway. So you can make this go faster. You could put inhibition in here. There's a lot of stuff that can happen, right? And so again, you have a lot of places where you can tweak the response to this, don't you? For better or worse. All right. Again, this is that part of the pathway that we just talked about. And so again, we have the G alpha with substrate right here, right? With the GTP bound. So after the GTP hydrolysis and release of the phosphate, which is right here, this hydrolysis, release of phosphate, we're going to have the GDP bound one. The G alpha is going to reassociate with the G beta and gamma. Remember that this gamma portion has a anchor on the cell membrane. So they're both wandering around on the cell membrane hitting each other. Okay? And this is a fluid mosaic, right? Mosaic fluid dynamic. Mosaic fluid. Okay? So it's rearranging and hitting each other and bang, bang. Okay. Okay, so this is going to then reform the inactive heterotrimeric protein. This inactive heterotrimeric protein can end up wandering back over to our 7TM, rebinding onto it, and it could actually end up getting reactivated, can't it? Okay? But it will require GTP. It did reset. There is a chance it's going to stop firing. Okay? As long as that epinephrine doesn't keep jumping on to the 7TM, it'll stop firing. Right? Or God help you if you get one of the ones that are there's substrates that we can put that will continuously make this fire, right? That binds better than normal epinephrine. Yeah. Anyways, termination of the beta-androgenic receptor signaling. So resetting the hormone-bound activated receptor to prevent the continuous activation of G proteins is going to occur via two processes. There's the hormone dissociation, which I kind of mentioned a little bit before. I got ahead of myself, I guess. The G protein receptor kinase phosphorylates the serin-3 residues in its receptors. Then the beta-arrestins bind and block the receptor's ability to activate G proteins. Okay, so we have the G protein receptor kinases. Okay, what do you think those G protein receptor kinases do? They're kinases that activate on the G proteins, right? Okay, specifically on the receptors of the G proteins, to be very specific. Again, this is going to phosphorylate your serin-3 residues in those receptors. Your beta-arrestins, arrestins have a great name, by the way. Arresting meaning what? Stop. Yeah, so you stop it. Okay? Beta, there we go. What was our receptor called again? Beta-androgenic receptor. The naming is actually really well done. Makes life so much easier to deal with, right? Okay. Are going to bind. They're going to block the receptor's ability to activate the G proteins then. Okay. So you have several things going on. You have phosphorylation that deactivates. You have blockers that are going to compete with it, right? And so you can further modulate that. This right here is actually the beta-androgenic receptor signaling terminated by your two different processes. So which one's the one signaling? Left, middle, right? Middle. Got it. That's the one signaling, okay? And so you have two different processes. You have going left, the dissociation. So if this is epinephrine, which is the one we've been talking about, right? It dissociates. And, you know, that happens all the time, right? And so what will hopefully happen is this gets washed away with your blood. You don't have a lot of more epinephrine coming in. And so then that drifts off probably into your kidneys and gets butchered apart, right? Or liver where it gets broken apart into scrap for you to use. And you don't have any more epinephrine coming that can bind it, okay? Or at least small enough that it keeps dissociating as its primary. This right here would be the phosphorylation pathway. So this is where you have so much epinephrine that you need to block this off, okay? It's too much. It's been going too long. You got a problem. And so this is going to be the phosphorylation pathway. Your epinephrine, or whatever it is binding to your 7-TM, and the receptor kinase is going to come along. It's going to use ATP to going to ADP to give you a phosphorylation of this receptor. Okay? So that's your receptor kinase right there. This phosphorylation is going to allow the binding of your B-arrestin, in which case it's blocking now that G protein from being able to get there, right? And so we have fully blocked it off. No more G proteins going. No more signal coming out. Okay? By the way, does anybody know if any of you have experienced this? You ever been stressed out? Yeah. You've likely experienced this. Okay? If you're stressed, you're going to slowly leak out more epinephrine than you should. And you'll almost be in a constant fight and flight response. And that's the stress response. That's why you get sick. That's why your immune system's weakened. That's why you have all these problems. Okay? And so this is actually ends up happening in the long term is you actually start deactivating a bunch of these 7-TM receptors and you stop responding to your epinephrine as much. You develop resistance against it. Yeah. Anyways, interesting stuff. Huh? Kinda, except when when you need it. Yeah, so, but no, honestly, you should read up on some of the literature. Like the stress response actually is just amazingly widespread problems to you. Anyways, some of the 17 receptors are going to activate the phosphoinositol cascade. So the alpha androgenic receptors are going to be receptors that activate the G alpha Q in this case. Notice the Q instead of the S. Okay? Okay, so the G protein, this binds to and activates the enzyme phospholipase C when in its GTP form. What's our time look like? 12:35? 25? Oh, okay. You take care. Have a wonderful day. So notice that we have phospholipase C when it's GTP form. This is different than the previous one, okay? So this is then expressed in your muscle cells that control the pupil dilation, and they line your small blood vessels in your skin. This will bind your epinephrine. It's homologous to the beta-androgenic receptors with very similar 3D structures. These 7 TMs, they are extremely similar. The secondary messengers are really almost all of those lists. Remember that small list I gave you? They're almost all of those, okay? And it widely just depends on the specific G unit, which one comes next, and it widely depends on the specific cell on what actually happens to that, okay? Okay, so this is the phospholipase C. Phospholipase C is going to catalyze your cleavage of your PIP2. This is your PIP2, by the way. This is going to be your cell membrane phospholipid. Notice it's the 2 different portions. You have your DAG, and you have what will be your IP3. But at this point, it doesn't have those 3 phosphates. They are not the same, are they? Because one is part of a phospholipid. Okay? By the way, notice hydrophobic tail, glycerol head, phosphate on that head, sugar on that phosphate. It's the same damn thing as we've seen all over the damn place, right? There's no real differences. It's just in this case you have an I, and it's going to have more phosphates on it, and cleavage is going to cause secondary effects. Okay? Okay, so your phospholipase C, this is what it's going to go through. It's going to cleave this, and this is actually going to make the IP3 and DAG. Is everybody okay with that? Okay, did I lose anybody in that? Okay, so we basically have the same thing going on up to G alpha, in this case it's alpha Q, which is going to go off just like it did before, but now it's going to activate phospholipase C. Phospholipase C is going to cleave DAG from IP3. Before it cleaves it, this is called PIP2. Does anybody need me to say the name for it? Because you're never going to hear the name again. It will always be PIP2 and IP3 and DAG. Okay? If you ever actually need the actual chemical name because you're doing research or something, just look it up. I'll be honest. I don't remember. And I've done research on DAG, which is really sad. I've literally done research on these too. And I'll be honest, I oftentimes will forget this name, the inositol portion specifically. Okay, does anybody have any questions on this portion of it? Which part's going to stay on the membrane? Is DAG going to go off into the cell cytoplasm and activate something? Yeah. DAG's going to stay on the membrane. It's going to float around on that membrane, right? It could activate stuff on the membrane, but it's going to be on the membrane, right? How about this one? Where's that going? IP3? Yeah, it's going to float off, right, into the water, because that's pretty dang hydrophilic, right? By the way, one of the easy ways to remember this is that IP3, all 3 phosphates are the same. That's why it's 3, right? PIP2, one of the phosphates has to attach to DAG. So the phosphates are different. Okay? Okay. All right, so this is IP3. IP3 is a soluble secondary messenger. This is going to diffuse away from your membrane. It's going to cause rapid release of calcium 2+ from the ER through a specific IP3 gated calcium 2+ channel protein. Notice again, that's another one of those secondary messengers we had talked about. Note, by the way, that the secondary messengers also are typically tertiary messengers and quaternary messengers. It just means it's not primary, right? Okay. The calcium 2+ is a signaling molecule. This is going to bind camulodolin, typically abbreviated CAM, capital C, lowercase a, capital M. This is ubiquitous signaling protein. It's going to bind enzymes such as protein kinase C. Yes, that is, again, PKC, right? Protein kinase C. Okay. So this is the DAG right here. DAG is going to, again, be a secondary messenger. This is going to remain in your plasma membrane, anchored there. It's going to bind and activate the protein kinase C. Again, that's that same PKC. Yes, these both work together on the same thing. Okay? So you see that this is wrapping around on itself, and they're both ending up on PKC. Okay, and protein kinase C, PKC, this is going to be a protein kinase that's going to phosphorylate serine 3 residues in target proteins. I'm sorry that the definitions are always the same on these things, but that's what a kinase does, okay? That's why I introduced the classes first, because PKA, PKZ, so it's the same damn definition, right? Again, the big difference here is this is going to require calcium 2+ to bind the DAG, which does help you with the naming a little bit, because PKC requires what? A C, calcium 2+ to bind your DAG. Okay, this is what this looks like. This is the cascade that's going to result in your protein kinase C activation, your PKC activation. So what we're seeing here is, again, this at this point is just your cell membrane. We have the PIP2. We're going to cleave it with the phospholipase C. We have already talked about the part that has the 7-TM receptor and the G protein, right? So that's happened, okay? So we've activated phospholipase C. This cleaves the PIP2. It's going to cleave it into two parts. One part is going to be your DAG, right? The other part is IP3. Your IP3s are actually going to go off and go to an IP3 receptor. Those IP3 receptors require 4 IP3s. This will then open the calcium channel on your ER membrane. Calcium will start going into your cytoplasm. Okay? So calcium is going to go up here. The DAG is already released and is floating along your cell membrane, right? The inside of your cell membrane. Okay. So your DAG is going to bind, 2 of them specifically, to your protein kinase C. Your calcium has to actually bind first typically. Okay? To activate this, your DAG can finish binding. Okay. Does anybody have any questions about that so far? Okay, so once you've bound these 2 components, you're going to have an activated protein kinase C right here. And this is going to start phosphorylating various different serine 3 residues. These phosphorylations, depending on the exact receptor cell combination, is going to cause things to activate or deactivate, which will then cause overall effects, right? Again, we're trying to go for a class right now. So your calcium ion is going to be a widely used secondary messenger again. The calcium 2+ is going to participate in many signaling processes. Some of these reasons is that fleeting changes in calcium 2+ concentration are readily detected. Calcium 2 plus can bind tightly to proteins and induce substantial structural rearrangements. Note that a lot of the protein activation sites require like 2 plus charge things, right? Because 2 plus charge things bind much tighter than 1 plus. Okay? And that's why you typically see a lot of these calciums are major binding components that will activate or deactivate something. Okay? Magnesium would also be that, but magnesium's too important to screw around with too much. Okay? All right, so this right here is going to be a specially designed dye. We can be used to visualize changes in your calcium 2 plus concentration. Specifically, this is going to be a fluorescent component, which is yellow. So we have this specifically designed dyes. We can have Fura-2, for example. This is going to bind calcium and change their fluorescent properties on binding that calcium. And so this is actually your Fura-2 right here, specifically. Okay. And so what we see here is we have the dye, and then we free cation concentrations inside our cell. And we can then image these using fluorescence microscopy, again, with our appropriate set of dyes. And so at time 0, what do we see? Nothing. Yeah, we haven't had any release yet. And so as our time goes on, what are we going to get? We're going to get more and more color, and then eventually it's going to fade back, right? Because we'll have our signal start tapering back off. Okay? Okay. Notice that it kind of blurs out at this point because we've fully calciumed the whole damn cell, right? Yeah. Okay, so this actually, the calcium ion is often going to activate the regulatory protein camlodulin. This is oftentimes referred to as the EF hands. So calcium 2 plus will bind, has this binding motif that actually binds in this camlodulin, is going to have this hand motif, the EF hand motif. And this is going to consist of a helix, a loop, and a second helix. So helix, loop, second helix. Okay? And you can notice that it looks like you're holding something with your hand like this, right? Okay. Kind of rests in here, and then you close over it. Okay. And that's why it's the EF hand. Calmodulin is going to be a common calcium 2 plus sensor in your eukaryotic cells. It's going to contain 4 EF hand motifs. Each of those can bind to a single calcium. And so right here is where we're actually seeing this. So again, we have here the calcium 2 plus binding to our calmodulin. It's going to induce a substantial conformational change in its EF hands. It's going to actually expose the hydrophobic surfaces that can then bind the proteins. And so right here we can actually see these EF domains stretched out. This is what it looks like without binding any calcium. Okay? And so now we have it binding, and this is going to be the calmodulin sensitive kinase right here, and that's the calciums right there bound to the calmodulin. Okay? And again, these are the hydrophobic domains down here. Okay, so some of your receptors are going to signal through G proteins that inhibit rather than stimulate adenylate cyclase. So before we had stimulated it, now we're going to inhibit it instead. All right, so this is your alpha-2 androgenic receptors. Again, this is going to be a class of 17 receptors. They'll signal through the alpha I. I for what? Or inhibition specifically, right? So these are going to be ones that will inhibit. They'll inhibit our adenylate cyclase. Excuse me. And so again, this is going to be the G protein that will bind and inhibit adenylate cyclase expressed by some blood vessels to smooth muscle cells. The activation of this pathway is going to result in a decrease of CMP rather than an increase in CMP. And so right here we actually see the alpha 2. Notice, by the way, the alpha 2 being the one that's the inhibition. The alpha 1 was the what? Just the alpha was? The activation one, right? Everything else the same. What time is it? Oh, we'll stop here then, since we're at the start of this. We can talk about it next time. Okay, I'll see you all Friday. You all have a wonderful day.